Hi, today we're doing the Human Microbiome Project, the HMP, and how it's changed our understanding of our normal biota. So, what is a microbiome? Your microbiome are trillions of microorganisms that are all over your body all the time. They're all good, they're bad, they're all there. You just don't know they're there. They're so small and so tiny. They're your first defense in protecting your body from all of the invaders and foreign invaders that try to damage and hurt your body. So we're going to break this down. So the United States Institute of Health, they did a 10-year study and they were trying to figure out what is the human microbiome and what does it do to us and for us and how does it help us. And so they wanted to understand it, and it was a very long process for 10 years. And they just wanted to find out disease-wise, what does it do? Does it help us? Does it hinder us? And they came up with all kinds of fun answers. And your normal biota, yeah, I figured I'd explain that too because I actually had to look that up. That is what makes you you. They are all your microorganisms that are present on you. That's you. So this is a little video that I got here that I'll play for you. Maybe. There it goes.
Our bodies are home to around 100 trillion microbes, mostly bacteria, inside and out. They outnumber our cells 10 to 1. This is the microbiome, and we couldn't live without it. There are diverse, complex communities of microbes in the intestines, in the mouth, on the skin, and elsewhere, living in a delicate balance with our bodies. Mayo Clinic is seeking to understand how the microbiome promotes wellness and enables disease, and how that knowledge can change the practice of medicine. For example, we know the microbiome interacts with cells in the lining of our intestines, helping them to absorb nutrients and energy, strengthening our immune system, and more. When this healthy community gets tipped out of balance, certain types of bacteria can grow out of control, causing illness. Until recently, the standard medical approach was to give antibiotics to kill the problem bacteria. But now we know that destroying bacteria further disrupts the balance in the microbiome, sometimes causing more serious problems. For example, one type of bacteria can cause a colon infection when the microbiome has been disrupted. Uncontrolled, it can cause severe tissue damage and death. Scientists at Mayo Clinic are finding new ways to keep the microbiome healthy, repair it, and use its powers to prevent and treat disease. Currently, researchers are studying the use of probiotics, beneficial microbes to treat problems in the gastrointestinal system. And Mayo physicians have actually cured certain gastrointestinal infections by transplanting a healthy microbiome from one person to another. Mayo researchers are also exploring the genetic code of the microbiome. Each microbiome has its own DNA, entirely different from the body's DNA. Techniques reveal which bacteria are present and help identify genetic markers linked to specific diseases. Mayo scientists are also using the latest techniques to capture highly valuable data about the microbiome genome and the metabolites they produce. This new information allows physicians to make early diagnoses and design new and more effective therapies, including preventive therapies. Researchers at Mayo Clinic are studying the microbiome in a completely new way and just beginning to understand how the genes of the microbiome can affect reproductive health, cancer development, and neurologic disorders. Find out how you can help Mayo Clinic bring the promise of microbiome research to life for every patient, every day.
So that was a nice little video. So I just have a couple of things here about the video. So what are the human cell? What is the human cell to microbiome ratio? Well, 1 to 10. So every human cell, there's 10 microbiomes. So we are severely outnumbered here. And then again, with the how many microbes do you have all over your body, in and on you? That was 100 trillion. So on the grand scheme of things, we're not really humans. We're not really people. We're just a bunch of and bacteria walking around in water. Don't forget to take your probiotics. So we need this for our overall health. We need this for everything. All of our daily health, everything we need them for. No matter what we think, if we think we're healthy, there's a microbiome that's involved in it. Somehow, some way. It goes from everything from our cardiovascular health to diabetes, asthma, Crohn's, autism, GERD, IBS, and even our moods. We can be moody just because of our little microbiomes. Everything is linked to our human microbiome. Even our digestion. Digestion's a big one. They really, that's the one that we seem to know the most about. So that's kind of where we began with this process. It turns everything, they make enzymes, and that helps us break down our food. So that's a big one. And it's more than we thought too. That, like I said, it was a 10-year study. This is what they figured out, is that you can't grow it in a lab. So that was the big breakthrough. Is up until now we've used the 5 eyes and we've come up with results where now we're finding that even if you don't grow it in a lab, it's still there. It exists. So if you can't grow it in a lab, it still exists. So this project was a big one, explaining that one. And here's another man named Rob Knight. He was involved in the microbiome project, and he's going to talk a little bit about it eventually, if my internet works. So.
We're talking with Rob Knight. Rob is a professor in chemistry and biochemistry and also in computer sciences at the University of Colorado. Rob, you're one of the gurus of the microbiome project, and we've learned so many new and different exciting things from that project. What are some of the real key surprises?
Well, I think one of the key surprises is how different we are from one another microbially. So, as you know, we're all essentially the same in terms of our human genome, 99.9+% identical. Whereas in terms of the microbes that we carry on our hands, in our guts, and on and inside different parts of our body, we're often 80 or 90% different. So it's just fascinating how much variation there is, and then one of the key questions is, does all that variation matter, or does all that variation not matter? Is it different because it actually makes a difference to health, or is it different because it can just vary randomly? Another fascinating thing is that there does not seem to be a core of microbes that we all share, but rather any microbe that you find as being abundant in one person, you'll find some other person who completely lacks it, or at least has it at such a low level that we can't detect it with the techniques that we usually use. So again, it doesn't seem like we're all colonized in the gut, at least, with the same stuff. You see very different species assemblages in different people. The third thing that I think is fascinating is the links that are starting to come up between microbes in the gut and the brain. So there's been some very interesting research recently on probiotics and anxiety in mice. There was also some work we did with Andrew Gowitz's group a couple of years ago, looking at how you can have a genetic mutation in the mouse that triggers a change in the microbial community that makes that mouse obese. But the way it does it isn't by making it more efficient at metabolizing what it's eating. What it does instead is it makes the mouse hungrier, right? So it becomes obese because it eats more. And you can cure it. You can cure it with antibiotics. You can also take those microbes and stick them in a completely genetically normal mouse with no microbes of its own, and that other mouse will also eat more and will also become obese. So, that's completely a behavioural thing. So I think one thing that's fascinating is just all of these connections where microbes may be doing things that we never suspected.
Yeah. There's more of them on us than human cells, and they do more things than we could have ever imagined. So one of the kind of grand goals that was often spoken of with the Microbiome Project is that, in fact, we would be able to develop probiotics that would influence a lot of these different aspects. But when we're so different, is that a realistic concept?
Well, that's a great question. So there are some kinds of therapies that have been extraordinarily effective, like stool transplants, for example, where you have people with Clostridium difficile that's so severe that it's resisted antibiotic therapies even for a couple of years, and yet the cure rate, just taking a stool sample, making a smoothie out of it, or going in the other route as well sometimes, and then transplanting a normal microbiota has been enormously successful, so 90 to 95% cure rates. So there are some kinds of things where it seems like these ecosystem level therapies are extremely plausible and extremely robust. So there's three things that we need to know in order to do therapies based on the microbiome, right? We need to know what a good community looks like. We need to know what a bad community looks like, and we need to know how to get from the bad community to the good community. And until recently, we haven't known any of those things, and now we know one of them, right? We know what a good community looks like, and so being able to figure out not just what the bad community looks like, but also how to get to the right place from there, I think is going to be really critical.
So you're from New Zealand, and New Zealand was one of the places that was so plagued by invasive species, where somebody said, I'm going to bring this species in because it's going to do something good for the environment, and in the end it was horrible, right? And the worry with modulating the microbial flora is that in fact maybe we don't know enough to be able to avoid those kinds of problems.
Yeah, it's something I worry about a lot, and developing the kind of sound ecological theory that allows us to predict what will happen when you introduce a microbe into a particular environment. I think we have a much better chance of doing that with microbes than was ever the case for macroorganisms, because they're so small that you can get you can do enough replicate experiments to really figure out if you can predict what will happen when you introduce a particular microbial member into a community.
So one of the sayings people use commonly is you can't see the forest for the trees. One of the problems when you have complex systems like this is sometimes you can't see the trees for the forest. So if you make estimates, how much of the diversity are we missing out there when you, for example, use 16S ribosomal RNA as a mechanism to try to identify who's out there?
Right. Well, so as you know, there's this thing called the Great Plate Count Anomaly, which basically says that of the things you can see under a microscope, only about 1% of them will actually grow when you try to culture them. And that's often confused with another number, which is what fraction of the kinds of species that you see, or rather a fraction of the kinds of sequences that you see with 16S sequencing, will you be able to grow in the lab when you culture from the same environment. So for some environments, we're getting really good at doing the culturing. So in the gut, for example, the discovery that a lot of gut microbes were microaerophiles rather than being strictly anaerobic or strictly aerophilic turned out to be really important for getting a lot more to grow in culture. And so the human gut, at least the gut of healthy young adults, that's getting pretty well tapped out. And so when we sequence a lot more, we're not finding a lot of new stuff there. Whereas there's other environments, like for example coal beds and other hydrocarbon contaminated environments, where you do the sequencing there and you find a whole lot of new diversity that's not even closely related to anything in the database. In the Earth Microbiome Project, when we map against green genes, which is this amazing phylogenetic resource compiling 16S from the database of different environmental projects, at the 97% identity level, which is often used as a rough approximation for species, we have already covered 82% of all of the species level OTUs that have ever been previously observed in the environment. So in the EMP, we're already doing pretty well. But in contrast, only about 17% of the tree is covered by things on the human body.
So that was a really awesome video. I really enjoyed listening to him. And then we're going to review a little bit here about the video. We're going to just say, like, what were some of the key surprises that he had mentioned? He had mentioned that they were awfully surprised when they found out each person was so different. You can have a same person, you can have a family of people, and each person would be drastically different microbiome-wise. It's pretty incredible that you, like the same issue we're having now with the coronavirus, where one family member will get it and the other people won't, and nobody knows why. So this is playing in with the microbiome, because each of us is different, even if we've been exposed to the same things everybody else have. Another one was the skinny mouse, fat mouse, obese mouse. What was he talking about with that? So C. diff also played in with that. What was he doing to these mice then to make them obese or skinny? It was poop smoothies, or lavage, basically. What I thought was pretty incredible, that he was taking an obese mouse and taking poop from the obese mouse and making a little poop smoothie for the skinny mouse, and then all of a sudden the skinny mouse started getting bigger and bigger and became obese. And they're finding out that it's actually a microbiome that's making them more hungry, not necessarily the fact that they're digesting things better or doing things differently. They're just hungrier, so they just keep eating, which I thought was pretty amazing for my diet. And then you have three main ideas about your microbiome, and that's kind of what they're trying to figure out. Is they're trying to figure out 3 steps. And the first step is you need to understand the good microbiome, you need to understand the bad microbiome, and then you need to know how to get from the good to the bad. So that's pretty amazing that they're now putting it together, and they're coming up with different ways to turn a bad microbiome into a good microbiome. Everybody has them. Everybody has them, and they need them. They are required for our life and our health, and they have good ones and we have bad ones. So they're constantly keeping each other in check. You need the balance between the bad and the good at all times, or one will overtake the other one, as we had mentioned earlier with C. diff. This is another little video this was my favorite one so far.
A dozen or so years ago, they published the sequence of the human genome, and those of us who are microbiologists were saying, they only got just a really small smidge of the human body, the human genome, and that the rest of it is the human microbiome, which is a collection of organisms that outnumbers the human cells by 100 to 1. We like to think of it as a community, and that there are these communities of bacteria all over the human body. So, our skin, our mouth, our gut are all covered in bacteria, and instead of being grossed out by this, people need to be excited about that, because these are the organisms that keep us healthy. So the Human Microbiome Project goal was to characterize the composition and function of the bacteria that live on and in the human body, and then to understand how changes in that community of bacteria affects human health. The main part of the project was to sample 300 people, 150 people from Houston and 150 people from St. Louis at 2 or 3 times over the course of 2 years, and they sampled them at 18 different parts of their body. And so the analysis was really, to some extent, crowdsourced to the scientific community to analyze. One of the really cool things about the microbiome in this study was that these people were very healthy, but they were all very different from each other. And so one of the cool things about the human microbiome is the community that you have really on any body site is your community. And that we think it's kind of the sum total of your genetics, your diet, your behaviors, how you were born, how you were fed as an infant, and that will stay with you forever. And to the point where my community is more like I will be 10 years from now than I will ever be like anybody else. And so in many ways, it really is a fingerprint. There's a European consortium called MetaHit that a couple years ago published a paper looking at the gut communities. And so what I thought was, well, the MetaHit study studied just the gut, and we had these 18 body sites. So I wanted to know is if we looked at the types of bacteria across these 18 body sites, do we see different types across the body? Are they similar to each other across the body? And then can I explain why I might be gut community type A and you might be gut community type B? And so this ability to categorize people into different community types for any body site, I think it's going to be a very powerful tool for understanding health and disease. And again, this is kind of one of the first steps in moving down that path. Previous work had shown that the bacteria in our mouth are very different than the bacteria in our gut. And so one of the things we found by assigning people's communities to these community types was that even though the communities and the types of bacteria are very different, the type of bacteria you have in your mouth can predict the type of bacteria you have in your gut. And so to me, this is exciting because it suggests that, if we want to manipulate the gut, perhaps we can manipulate the mouth. And that there's some data coming from various other labs unrelated to this project, finding in people with colon cancer high levels of bacteria that are known to be pathogenic and common in periodontal disease. And so if we better understood how those bacteria get to the gut, or again, how we can manipulate the mouth to manipulate the gut, that might be pretty cool too. One of the ways that this study has helped advance the field of microbiome research is this idea that the bacterial communities that are found across the human body can be associated with each other. Again, the mouth being connected to the gut or your left and right arm, and that we can relate those to kind of your life history. And then going forward, what my lab is interested in, and others at the University of Michigan and others around the world, is are there certain community types at different body sites that we can relate to health? One of the great things about where we're at the University of Michigan is that there's a real big push now to study the human microbiome. And so within the School of Medicine, we have the Host Microbiome Initiative, where there's a large number of faculty, 50 or so, interested in the role of bacteria in maintaining health. It's a really exciting time to be at the University of Michigan because we've got a lot going on and there's a lot of excitement in this area of research, both within the biomedical world as well as across campus.
So that was my favorite video. So basically what he was saying, it's only the beginning. We are in the absolute infancy stages of understanding the microbes and how they work and everything that's going on with them. So he was basically talking about change your microbes in your mouth and you can change your microbes in your gut. So that being said, can we figure out colon cancer? Can we change if we're going to get colon cancer or not? And he made it sound like it's a possibility. If we can change the microbes in our mouth, then we could possibly change whether or not we get colon cancer. Brush your teeth, I guess. I don't know. So, what about your right and your left hand? Do you have different microbiomes on your left hand and your right hand? Yes. Yes, you do. If you're right-handed, you'll have different ones than on your left. And your left will have different ones. Depends on which hand is dominant as to what you get on what hand. So I thought that was kind of cool. And then when he mentioned a little bit about being born and what you ate as a child and stuff, that was also another study where they were saying that being born naturally versus being born C-section was a big thing because you got all the different microbiomes from being born naturally. And then breast milk was another big one, is if you were fed breast milk versus formula, and that creates a total different normal biota for you as an infant, and that carries on through for your entire life. It changes everything. So there are studies out about that. So it's basically, we are at the very tippy top of the iceberg on this one with the understanding all of this. And they've all been talking about C. diff. So that's a big one. So C. diff is basically when you're in the hospital and you're on broad spectrum antibiotics, you've been on them for long term, you've been in the hospital for a long time, you're really getting lots and lots of antibiotics. So they wipe out all your colonies. And like we said earlier, balance is the key. You have to have a balance with your microbiome, and because if it gets out of whack, then all of a sudden the bad ones start growing, and that's where you end up with C. diff. And nobody wants C. diff. It is not something you want. So that is a big one. And this was also what they were talking about when they were talking about the poop smoothies or lavages. They were saying that basically, I think it was a 90 to 95% cure rate with the microbiomes if you did poop smoothies or lavages of a healthy microbiome. And they were saying it was pretty much curing C. diff. Well, I like this little graph here. I just like the fact that it did the 10 to 1 again with your human microbes outnumbering your microbiomes outnumbering your human cells. And I didn't realize your GI tract could cover 2 tennis courts. I didn't realize you had that much GI tract in there. And okay, so now we have a couple questions. Does the bacteria of microorganisms affect your overall health? Does the balance? So we've been talking about balance. We've been saying when we said C. diff, we said balance is the key, and everything we've been saying balance is the key. So I'm going to click true here, and true is correct. So we're going to go back, and then we're going to go, can antibiotics cause dyssymbiosis? And that is true. So dyssymbiosis is like what we were talking earlier. That's the technical term for when everything's all out of whack and nothing is the way it belongs. And this Clostridium difficile, that's C. diff. Is it always in your gut? Do we always walk around, healthy or not, with C. diff? And that is also true, because like we had mentioned earlier, where we'd said that the balance is what you need. Balance and balance and balance. We're going to keep saying balance. So basically, it's always there. And if you mess up the balance with all the antibiotics, then your balance is off. And then the C. diff starts growing, taking things over. Next, can we always use the 5 eyes to determine what microorganisms are present? We had mentioned earlier about the 5 eyes and talking about growing things in the lab, because that was what we thought up until now, was you do the 5 eyes, you grow things in the lab, and it's true. Well, this microbiome project proved otherwise. So can we always grow it in the lab? No, you can't always grow everything in the lab, and it's still there. So just a little review. Is your microbiome health important? Yes, extremely important. We need it for everything. We need it for everything we do. Has our understanding of the microbiome changed recently? Yes, they did a huge study with the past 10 years, and the reason that it's really become very popular today is because we are getting more and more diseases and health issues that are becoming resistant to antibiotics. So we're trying to come up with other ways to combat these issues, like the poop smoothies with C. diff. Are all bacteria bad? No, not all bacteria are bad. You have them on you all the time, good and bad. And would we survive without it? Would we survive without our microbiome? Nope, we would not. We need it, and we must keep it healthy. So take your probiotics. And there's my sources. Thank you.